What if you could read your own instruction manual? I mean, the complete biological blueprint that makes you, well, you. For decades, reading this book of life, our DNA, was pretty much the ultimate quest in biology. So today, we're going to dive into the revolutionary technology that finally let us crack the code.
So this incredible molecule, DNA, it's tucked inside almost every single cell in your body. And the best way to think of it is as a code, a really, really long one, about 3 billion letters long to be exact. For the longest time, the idea of reading that entire code from start to finish, that was pure science fiction. It just seemed like an impossible task.
And for a while there, it really was basically impossible. This number, $3 billion, that was the staggering price tag for sequencing the very first complete human genome. I mean, it was a monumental achievement for sure, but at that cost, it was a scientific marvel, not a practical tool that could change everyday medicine. Something had to give.
Okay, so let's start with Chapter 1 of this story, that first massive effort, which we now know as the Human Genome Project.
And this thing was a true marathon. It was a global collaboration that kicked off way back in 1990 and took a full 14 years to cross the finish line. A milestone for humanity? Absolutely. But it was also incredibly slow and, as we just saw, fantastically expensive. This wasn't going to be the future.
So what if you could take that 14-year timeline and shrink it down to a single day? A technological breakthrough is about to come along and ignite a full-blown sequencing revolution. This slide really shows you the big shift. Over on the left, you've got the old Sanger method. You can think of it like a master calligrapher. It produced these beautiful, high-quality long reads of DNA, but it was painfully slow. On the right, you have the game changer, next-generation sequencing, or NGS. Its strategy is just brute force. Instead of reading one long strand, it reads millions of short fragments all at once. The trade-off? Each individual piece is maybe a little less perfect, but the whole process is unbelievably fast and, crucially, super cheap.
And this chart, wow. This brilliantly illustrates just how fast we're talking. Just look at that curve. For decades, sequencing output was kind of growing at a steady pace. Then, around 2005, NGS hits the scene, and the amount of data we could generate per day just goes vertical. It was a complete explosion of information.
Now, what's really wild is what happened to the cost. That white line there, that's Moore's law. You know, the famous rule for how quickly computer chips get cheaper and more powerful. The green line is the cost to sequence a human genome. It didn't just follow Moore's law, it completely shattered it. This right here is what made genomics accessible to everyone. So what was the secret sauce? What's the magic trick here? Well, as this slide says, it's massive molecular parallelization. All that really means in simple terms is doing millions of things at once. Instead of slowly reading one long piece of DNA, NGS reads millions of tiny fragments all at the exact same time. That is the core innovation.
So how does this whole massive parallelization thing actually work? The best way to think about it is like solving a high-speed biological jigsaw puzzle. It all happens in 3 basic steps. First, you don't even try to read the whole 3 billion letter book at once. You shred it. The DNA is fragmented into millions of tiny, easy-to-read pieces. Second, the sequencing machine reads all of those millions of pieces at the same time. That's the parallelization part. And then third, and this is the really hard part, a powerful computer acts like a puzzle master, finding where all the little pieces overlap and stitching them back together to reconstruct the original book.
And here's a closer look at that computer assembly step. What the computer does is it takes all those millions of short reads and aligns them against the reference map, sort of a master copy that we got from the original Human Genome Project. By seeing where each small puzzle piece fits onto that master map, it can rebuild your unique sequence piece by tiny piece. Now, when you can suddenly sequence an entire genome in a day, you create a whole new kind of problem. It's a good problem to have, but it's still a problem. An unprecedented flood of information that we call the Great Data Deluge. This created this fascinating shift in the scientific bottleneck. For decades, the hardest part was just generating the data in the lab. With NGS, that became the easy part. The new challenge, the new bottleneck, became analyzing all that data. The problem literally moved from the biologist's test tube to the computer scientist's server farm.
This chart sums it up perfectly. With NGS, we gained incredible speed and a really, really low cost for each letter of DNA we read. But the price we paid for all that progress was a new and extremely high demand for computation, for data storage, and for some pretty sophisticated analysis.
Okay, so we've got this incredibly powerful technology, but what does it actually do? Why does it matter out here in the real world? Let's take a look. Turns out we can do some pretty incredible things. We can diagnose rare genetic diseases in newborn babies. We can sequence a patient's tumor to pick the exact right cancer drug for them. During a pandemic, we can literally track a virus as it evolves in real time. We can even take a scoop of soil or a bit of seawater and sequence all the DNA in it to understand entire ecosystems. It's truly transforming medicine and biology.
So what are scientists actually looking for in all this data? Well, they're hunting for variations. And as you can see here, that could be something as tiny as a single-letter typo in the code, which is called a single nucleotide polymorphism, or a SNP. Or it could be massive structural changes where entire chunks of a chromosome are deleted or duplicated or moved around. These are the kinds of genetic variations that can lead to disease, and NGS is the high-powered magnifying glass that finally lets us find them.
But the story doesn't end here. Of course not. This revolution is still ongoing. So let's take a quick peek at what's in the next chapter for genetics. The next frontier is something called third-generation sequencing. And the goal here is to fix the biggest drawback of NGS, all those short little reads. The idea is to get the best of both worlds, the lightning speed, but with the high-quality, long reads of that original, slow method. By reading DNA in much longer stretches, that computational jigsaw puzzle becomes dramatically easier to solve.
And believe it or not, this isn't science fiction anymore. On the left, you see an animation of a technology called a nanopore, which pulls a single strand of DNA through a tiny hole to read it. And on the right, that's a real device. It's called the Minion. It's a handheld DNA sequencer that you can literally just plug into your laptop's USB port. The future is, well, it's already in our hands.
And this brings us to our final and maybe most critical thought. This technology is advancing at an absolutely incredible pace. We are rapidly approaching a world where sequencing your own genome could be as routine and as cheap as getting a blood test. So the question isn't just for scientists anymore, it's really for all of us. As this power becomes universal, how are we going to navigate the profound ethical challenges that come with being able to read the secrets hidden in our own book of life?